Hello student, how are you? I hope all of you are fine. Today we are going to discuss about the microbial cell culture and its application. In the previous lecture we are discussing the different things such as the requirements of for the cell culture and media about the cell culture, and in this lecture we are going to discuss the vessel that is used during the cell culture. It's a bioreactor. I have already told you it's a bioreactor is used for the culturing the microorganism in the industry. So we are going to discuss uh, in this lecture about the bioreactor. So now we comes on the introduction about bioreactor. Basically, bioreactor is the cylindrical vessel. It's used for the carried out the biological processes in the producing the or culturing the microorganism in the large scale. So we are culturing the microorganism in large scale in the industry. That's why we are using the bioreactor, and the bioreactor is also called the fermenter in the industry. So here what we do in the bioreactor. And bioreactor in the bioreactor we are in the last lecture I have told you already the different the commercial products such as the antibiotic, ethanol, recombinant products such as insulin, hepatitis B vaccine. All these things we have produced in the bioreactor. It's just a vessel, and you can compare the bioreactor with the flask that is using in the lab, and so and the bioreactor is used for the producing different type of product. So now it's clear. And the in the bioreactor there is some process is also occur, such as we can maintain the such outlet or inlet conditions so that we can add the media during the process, and fresh media during the process, and between the process we can drain out the substrate or product from the bioreactor. So it's a we can study these things in the structure of bioreactor. So what we producing in the bioreactor I have already told you, such as enzyme, by using enzyme, antibiotic, ethanol, by using the different cells such as microbial plant cell, we are also using in the bioreactor for producing different metabolites. Animal cell is also using for the different disease treatment and different point of view, and human cell is also using, or we can say the bioreactor is also used for the culturing plant cell, animal cell, and human cells. So in the bioreactor, there is some controlled, in the bioreactor, the control environment is required for culturing the microorganism or cells. So we are design the bioreactor like that. It can control the temperature, pH, substrate, salt condition, vitamins, oxygens. All these things are present in the control condition because we are know as the control condition are required for growing the cells in the biological process. At the optimum conditions, the maximum product is formed. So, on the basis of its structure and on the basis of which type of cells you want to grow, and different type of the bioreactor are present. But here we are discussing the main two type of bioreactor, such as simple stirrer tank bioreactor, sparger stirrer tank bioreactor. Yeah. So here the sparger is used for the elution point of view. So now we are going to discuss in the about the structure of bioreactor. You can see just it's a simple vessel, and in which the different structure is present, such as one motor. Yeah, so it's a main part, and the with the motor it's a axis, and there is the attachment of the impeller. Impeller is used for the mixing the nutrients, mixing of oxygen and mixing of nutrients. So it's an important part we can know. So it's an impeller question is asked the in the bioreactor, what is used for the mixing in nutrients. So it's a impeller. And next it's a pH controller. Yeah, so acid or base we can it's a sensor that sense the pH of the medium, because the microbes are grow at a optimum pH. So it's a pH sensor is attached with it, and the pressure sensor is also attached with it, and temperature sensor is also attached with it for the sensing, such as pH probe. Yeah, temperature probes. What is the probes here? These probes are the sensing the temperature and pH of the medium. Yeah, so and this is sparger that is used for the mixing. Here, this is in the base, you can see it. It's a sparger, and the oxygen is compressed oxygen is passed from that path, and this sparges by the sparger in the medium, and that is mixed by the impeller. So here the oxygen probe, it's a very important question is asked, what is used in the bioreactor for the optimum conditions, and or how to control the optimum conditions of medium for the growing the microorganism. So it's a pH control, temperature control. Different probes are used for the controlling it or checking it. It's a oxygen concentration probe. Yeah, so these are the things, these are the sensor that is attached with the bioreactor, and it's a impeller that is used for the mixing the substance in the bioreactor. So next is the most important, it's a inlet. From that nutrient or inoculant is added here. Clear? So this is the antifoam. From that inlet we are adding antifoam. In the last lecture we have also discussed what is the antifoaming agents, and the why it is required. Clear? So it foam is produce the hindrance for the producing the product. So that's why we are adding the antifoaming agent, different as sunflower oil, olive oil, and silicones is also used as an antifoaming agent. So now we can study the structure of the bioreactor. So most important things is the jacket. You can see here. In the last lecture I told you, for the sterilizing the media in the bioreactor, and the steam is passed through the jacket. Clear? So hot steam is passed in the jacket, and from the bottom it's a cold water is passed. Clear? And for the cooling the jacket, once we pass the steam, after that we passing the cold water for the cooling the of the medium, and so it's a very important structure of the for producing the microorganism in the or producing the product in the industry. So in the next we can see, this is the structure of the bioreactor. It's a glass bioreactor, and it's a stainless steel bioreactor. So it's a very small bioreactor that is used in the laboratory condition, but we are moving towards the industry. So there is a high capacity bioreactors are used in the industry. So, but structure is same. All thing is same here. Temperature, pressure controller, jacket, all these things is same. But it's a large bioreactor are used industry, because we are produce a high amount of the product in the industry. So next, how the bioreactor is function. So the bioreactor is function in the controlled condition. So it produces the cell biomass, metabolite by the plant cells is using for the producing metabolites, and other cells is also using for the producing metabolite. Transform products such as recombinant product, insulin, and hepatitis B vaccine, single cell proteins. All these things are produced by the bioreactor. So next, what is the required in the bioreactor? It's a agitation is required, oxygen transfer is required. I have already told you, once you transfer the oxygen, so it's a agitation is required. Clear? For the mixing the oxygen, temperature controller, foam controller, foam production, pH. All these are affecting the, all these factors are affecting the production of the substrate in the industry. So that's why we are maintaining these parameters in the bioreactor. So bioreactors should have following uh, qualities, such as it can easily sterilized. Once you have used bioreactor, so it's required to aseptic conditions. Yeah. So the bioreactors you have designed, it's easily sterilized. Yeah, aeration and agitation is properly present in the bioreactor. Power consumption, what is the power consumption? If the power consumption is high, then the cost of the bioreactor is increased. So as well as the maintenance of bioreactor is increased, as well as that affect the cost of your product. So that's why it's important. Power consumption, temperature control, pH control, sample facility is available, evaporation losses is not much high. So these are the factors. Next, minimum use of labor. If the more labor is required, so it increases your the eventually the cost of your product. So we are maintaining all these things such as contamination, aseptic operations. All these things we are internal smooth surface is also present so that it cannot harm your the growth of the microorganisms. So that's why all these things are required for the reducing the cost of the products. The cost is the very important factors for the bioreactor. So in the bioreactors, what is the control parameter is required? We have already discussed, such as the heat is produced during the process. So the heat controller is present. So what is the heat controller? It's a jacket. Yeah, so cooling jacket is present that in which we pass the water that is used for the controlling the temperature. Agitation, proper oxygen. Yeah, next is the homogenization of the media. Homogenization of the media should be in homogeneous condition so that microorganism can easily use this medium. Next is the foam controller. Clear? So foam is easily controlled in the bioreactor. So it's a very important parameter that is should be controlled during the fermentation process. So next, agitator, that is the impeller that is used for the agitation process. Baffles, aeration system, that is sparser, and we have already told you in the structure. And now we are coming on the application of the bioreactor, and the bioreactor is used in the fermentation industry, that is a bioprocesses. So the for the production of different products such as antibiotic, ethanol, I have already told in the previous lecture also. So these are the applications. And cell culture, bioreactor is used for the culturing the cells. Now, thank you so much for watching this video. I hope all of you understand. If you have any doubt, then ask me. And, thank you so much for